Hello, my name is Emily Ball, and this is my video lecture on the role of the pancreas and the liver in regulating digestion. I will start the lecture by going over the anatomy and function of the pancreas. The pancreas is this organ, seen here, in very close connection with the small intestine. And running through the center of the pancreas is the pancreatic duct. As you can see in the image in the top left corner, the pancreatic duct goes from the pancreas directly into the lumen of the small intestine. And this portion of the small intestine is known as the duodenum. This close proximity and relationship between the pancreas and the small intestine is essential to its role in digestion. So pancreatic cells, which are seen in the cross-section on the right-hand side of the screen, are two different types. There's endocrine cells and exocrine cells. The endocrine cells are not directly related to digestion in this sense, and their role is to secrete hormones directly into the circulatory system. The two main hormones we see secreted here by the endocrine cells of the pancreas, which are in this image, are the islet cells seen right here, are insulin and glucagon, and these are antagonistic hormones involved with the regulation of blood glucose levels. Now, in terms of digestion, the important cells of the pancreas are the exocrine cells. And they are seen in these formations, these sort of clusters, orange clusters here, and they're known as the acini cells. And they are the exocrine cells that secrete digestive enzymes. Traveling down these arrows, where we see the word lumen written, is the pancreatic duct. Now, in addition to the acini cells, this area is lined with duct cells. Duct cells secrete a watery solution containing sodium bicarbonate, and altogether, the watery solution from the duct cells and the enzymes from the acini cells are secreted from the pancreas through the pancreatic duct into the lumen of the duodenum and are essential to regulating digestion, which I will discuss next. So the image of the left-hand side shows the interaction between the pancreatic duct and the lumen of the small intestine. But it's first important to note how we cause this secretion of the pancreatic substances into the lumen of the small intestine. There's multiple stimuli that can cause this, including distension of the small intestine, food present in the intestine, neural signals that directly stimulate the pancreas, as well as CCK, which is a gastrointestinal hormone that enhances our recognition that our hunger has been satisfied after eating. So once the stimuli occurs and the pancreatic substances are secreted, they leave the pancreatic duct, which is seen in this image right here, and enter the lumen of the small intestine. This duct goes through these blue cells here, which are the intestinal mucosa, which do have a brush border on the apical surface. This brush border is made up of microvilli that increases the surface area of the apical surface, which is also an important function in digestion. And in yellow on the left-hand side, we see the substances secreted include trypsinogen and zymogens. Trypsinogen is a precursor to a digestive enzyme, which will be discussed in a moment, and the zymogens are inactive forms of the digestive enzymes that need to be activated in order to play a role in digestion. So the first step in this cascade is seen in the blue box overlying the intestinal mucosa, and that is the secretion of enteropeptidase from the intestinal mucosa cells. This enzyme activates the trypsinogen, which is the precursor, into trypsin. Trypsin is then an active enzyme, and its role is crucial in regulating digestion. It does this by activating the zymogens into active enzymes. Once activated, they're able to digest or break down the undigested food substances that's traveling in the chyme through the lumen of the small intestine. And that is how the pancreatic secretions are therefore regulating the digestion of food in the small intestine. So moving on to the liver. The liver is this large organ seen here in the right upper quadrant in this diagram in the upper left corner. It's actually the largest internal organ. When we zoom in this image on the right, we see that it is in very close association with the stomach, the gallbladder, this green structure here, and then from the stomach, the small intestine branches off. So the liver cells themselves are known as hepatocytes, and liver cells have many functions, one of which is directly involved with digestion. If we look in the lower left corner, we see what are called lobules, which is the hexagonal arrangement of the hepatocytes, and it's important to note that they are in very close proximity with the surrounding vasculature, which is essential to the filtration that occurs in the liver. In the upper right, that close association with the gallbladder is also essential in digestion, which will be discussed next with the production of bile salts. Bile is non-enzymatic, and it is secreted by the liver and is sent to the small intestine. However, it is sent to the gallbladder to be stored so that it can be released in appropriate quantities for fat digestion after larger meals. But the gallbladder itself is not essential to digestion. The bile salts, however, are, and they are enormously important in the digestion of fats, and this is because fat droplets, or lipids, are not water soluble. So we'll see in this image these large emulsions of fat droplets traveling through the lumen of the stomach, and they're too large to be digested, and their insolubility makes the digestion process difficult. Bile salts, one of the components in the bile secreted by the liver, is how this is accomplished. Bile salts have a hydrophobic side and a hydrophilic side. In the upper left corner, we see that the hydrophobic side connects with the water-insoluble lipid droplet, and the hydrophilic side remains in the water, which is present in the lumen of the small intestine. Hugely important to the digestion of the fat is making it water-soluble. The addition of bile salts to these large emulsions breaks them down into smaller, more reasonable size for digestion. This increases the surface area that allows the digestive enzymes from the pancreas to have more opportunity to break down these lipid droplets so that they can be reabsorbed or secreted as necessary. So after emulsion by the bile salts, pancreatic digestive enzymes break them into micelles, seen here, and then the much smaller, much higher surface area droplets that have now been formed through this process are able to cross the apical surface of the intestinal mucosa, which is this blue, slightly cut off in this image here. What's interesting about bile salts is that they are not damaged or ruined during this process, and they are instead recycled and can be used again for the addition of continued breakdown of fat emulsion and increasing surface area in order for the fat to be digested. And that concludes my lecture on how both the pancreas and the liver play key roles in the regulation of digestion.